Hello, Biolab 2. We are the Cactus group. I am Christian Amaro, and my team members are Ha, Phoenix, and Lewis. Our organism is Acinetobacter Baumannii, and we'll be presenting it shortly. Baumannii belongs to the family Moxella and the genus Acinetobacter. It is one of the 6 most important drug-resistant microorganisms in hospitals all over the world. It has large antibiotic-resistant islands and is known to rapidly develop resistances to antibiotics commonly used in hospitals and in war fronts all over the world.
Hello everyone, my name is Ha. I'm from the Cactus group, and today I would like to introduce to you about how the bacteria Acinetobacter Baumannii look like and what's gonna be their favorite places to live. So first, we can talk about how they look like. So the Acinetobacter Baumannii, they are gram-negative bacteria. So they have the rod shapes at birth, and so after that they transform to the spherical shapes at the growth phase. Therefore, they can be considered at the coccobacillus, which is in between the cocci shapes and the bacilli shape. They non-flagella and non-motile. So the names Acinetobacter is come from the Greek word akineto, stand for non-motile, and the full name of it, Acinetobacter baumannii, is named after Dr. Paul Baumann, who published the isolations of Acinetobacter from soil and water. So even though their names mention the non-motile ability, there are actually some gliding and tricking motilities have been recorded. Once again, these bacteria are naturally found in soil and water. They belong to the family Moraxilla, genus Acinetobacter, and the species Baumannii. So the pictures right here, as you can see, this is the Baumannii under the low power magnification of 10x using a digital Cajun scope. This photograph depicts the colonial growth displayed by the gram-negative Acinetobacter baumannii, which were cultures on the sheep blood agar medium for a 48-hour time period at the temperatures of 37 Celsius degree. Figure 3 is showing another view of Acinetobacter baumannii on sheep blood agar, but now with colors. So we can see that the colonies appear to be pale or white colors over the red media. And on the figure 4 is showing the same pathogens, but now it's in cultures on Markonki agar, in which the colonies have the light pink colors over the light yellow media. The study fields of Acinetobacter distributions and habitats is enormous, yes, very interesting. So we have the Acinetobacter genus is Arabid, and initially located in soil and water cultures, but the habitats of Acinetobacter baumannii is diverse, since they are ubiquitous and can live a prolonged period on the skin and inanimate objects regardless of dry or moist conditions. Several researchers have found that various of Acinetobacter baumannii generations can colonize in meat, vegetable, and fish, which is the food source for humans and animals. Therefore, Acinetobacter baumannii is also responsible for the widespread infection in pet clinics. And to human, they commonly aim at the patients who are the most vulnerable in the hospital environment. The colonies has been found on the patient's skins, respiratory tracts, gastrointestinal tracts, or even on the personal protective equipment, surgical tools, and respiratory care equipment. And with them, the infections have been critical issues all over England, United States, Argentina, Brazil, Hong Kong, Taiwan, China, Korea, and Japan. So the pictures right here is showing the area on the human body that Acinetobacter Baumannii can infect because they can follow our bloodstream when they enters. They can cause the pneumonia, infections of wound, or infection of respiratory tracts, infections of urinary tracts, even blood infections. I can say that I am impressed with these pathogens. It is hard to believe that just a little microbes like the Acinetobacter Baumannii, they can infect critical condition patients and develop resistant gene to many types of antibiotics. So as a result, they put their distributions into an international scale. Here is some examples of Acinetobacter distributions over the world. In the first pictures, we can see that every continent has reported Acinetobacter infection cases over the years. So the red colors right here indicate the outbreak report before 2006, and then the yellow colors indicates the outbreak since 2006. And the next one is to demonstrate a soldier who is in a critical condition because of the Acinetobacter Baumannii infections in the Middle East. So with the enormous number of the Middle East soldiers, especially in Iraq, who are infected by the Acinetobacter Baumannii, the researchers has to give it a nickname to the pathogens called the Arachnobacter.
Hi, my name is Luisa Pulvera, and I'll be covering the ecological importance section of this group presentation. An Acinetobacter is known to survive on hospitals and the equipment used in hospitals for long periods of time. Acinetobacter has been proven to build a resistance against antibiotics, which can lead to severe infections, and those severe infections typically affect critically ill patients in hospitals. With that said, Acinetobacter plays a crucial role in ecosystems as the Acinetobacter has developed at a rapid rate throughout all ecosystems and developed a resistance to multiple antimicrobials, which makes it prone to stay in environments for a longer time, and that affects the ecosystem in a larger way.
Hey y'all, my name is Phoenix, and today I will be introducing the nutrition and reproduction of this bacteria, which is called Acinetobacter Baumannii. So, a little background about this bacteria is that really, there's no really extensive type of research that could really give you anything on this bacteria because it's so weird. So, this bacteria, like they probably stated earlier, is found in soil. You can find it in meat. It's basically bacteria that's all around. You can get pneumonia from it. So this bacteria is found in hospitals, which you see, it's a hospital. So you're gonna find different bacteria, different diseases, all of that. So if you don't wash your hands, if you always touching surfaces, touching your face, hands in your mouth, you're gonna get sick. Period. So, Acinetobacter Baumannii is an auxotrophic bacteria, which means this requires a medium to be with the metabolite that they can't reproduce. So, in order to get this bacteria to reproduce, you have to transfer the DNA to a prototrophy stain. So prototrophic cells are considered a wild type of cell. So this means that these cells are self-sufficient producers of all required metabolites, amino acids, lipids, and stuff like that. So That's the only way they can confirm that is like an Acinetobacter bacteria. Now I will be going over, the virulence factors of. I had a moment. I'm gonna be going over the virulence factors of Baumannii. So, as you know, Baumannii, it hosts cell interactions. So, extracellular Baumannii experiences a metal-limited environment, at least in part due to metal binding host proteins and extracellular localizations of metals, which is red blood cells. Baumannii attaches to the host cell through interactions between fibronectin and bacterial outer protein membranes. So, like, for example, the protein A. So, it's also called maybe a type 5 collagen. These proteins may bind to other host cell proteins. For example, the interaction between the protein CHO-P with platelet activating factor receptors. Fimbriae also contributes to the eukaryotic cell adherence. Invasion into epitheliate cells occurs via host cell actin, filament, and microtubule reorganization. So recruitment of beta arrestin and quercetin and bacterial uptake via a zipper-like mechanism. So although Baumannii can resist intracellularly and includes entry into endosomes, the intercellular trafficking of Baumannii is not clear. Eukaryotic cell proteins NRAMP1, ZIP8, and ZNTS, and other transporter pumps essential metals out of the endocytic vesicles in cytoplasm, limiting the intercellular pool available to pathogens. This physiological relevance of intercellular life cycle Baumannii remains to be understood. One consequence of host cell exposure to Baumannii is apoptosis. It is possible that cell death or host cell invasion serve permanent Baumannii. I'm saying A like so. It serves permit to Acinetobacter Baumannii, and dissemination to deeper tissues, leading to an invasive disease. Okay. Okay, y'all. So now I'm going to be going over the reproduction of Acinetobacter Baumannii. So, Acinetobacter Baumannii is part of the phylum Proteobacteria, which means that this bacteria reproduces asexually through budding or binary fission. So, binary fission is like the most common reproduction for bacteria. So that means the cells divide into two similar daughter cells. So when the bacteria process begins, the two chromosomes of the bacteria get attached to the cell membrane and then replicate into bacterial chromosomes. So that means as the cell enlarges, the daughter chromosomes get separated. So as y'all could see over here, like this elongate, and then it causes, boom, see it's elongating, so it's like starting to replicate the cells. So as it enlarges, it causes the daughter cells to get separated, and it causes a cross wall. It forms a cross wall, and it divides the two daughter cells. So as y'all can see, as we get lower, it divides them completely, and it forms two daughter cells. So, as y'all see, this process could take about 20 to 30 minutes, I guess, depending on the bacteria. And sometimes the bacteria may only divide once, depending on the conditions of where it is. So, like in this case, Baumannii, it's grown in soil. Well, not grown, but it's found in soil and different surfaces. So I guess you could say not too hot of an environment, but like maybe like moist room temperature environments type stuff, if you get what I'm saying. So yeah, that's the reproduction on Acinetobacter Baumannii. And these are our references, which it cut off the name of the references. And that's it. Thank you for watching.